Welcome students. Myself Adshaya, lecturer of Emmanuel Arthur College of Pharmacy. Today we are going to see about consolidation parameters, diffusion and dissolution. Consolidation, an increase in mechanical strength of material resulting from particle to particle interactions is called as consolidation. An increase in mechanical strength of material resulting from particle to particle interaction is called as consolidation. Compaction equal to compression plus consolidation. Kinetic model study. First one, model dependent method. Second, model independent method. Model dependent method consists of zero order, first order, Higgs-Crowell law, Corsi-Meyer-Pippas model. Model independent method, pairwise procedure, F1 and F2 comparisons. Diffusion. Diffusion is the process of migration of solute molecules from region of high concentration to region of lower concentration. So, diffusion is the migration of solute. Solute means particles, samples from the region of higher concentration to the region of lower concentration. The passage of solute molecules through barrier mechanical by simple molecular diffusion, permeation, or by movement through pores and channels. The passage of solid molecules occurs through a barrier by simple molecular diffusion or by pores and channels. Fick's first law of diffusion. Fick's first law states that quantity of a solute diffusing through the unit cross section in the barrier in time d is termed as flux and is represented as J equal to dq by S dot dt. Flux is the rate of transfer of solute molecules per unit area of surface. It is the rate of transfer of solute molecules per unit area of the surface. It is proportional to concentration gradients. Flux is proportional to concentration gradients. It is given by J equal to minus d into dc by dx. C is the concentration, x is the distance of movement perpendicular to the surface of the barrier, d is the diffusion coefficient. The negative sign indicates that diffusion takes place in the direction of decreasing concentration. D is also affected by temperature, pressure, solvent properties, chemical nature of diffusing solute. Time lag. Before that, we see about Fick's second law. Fick's second law states that the change in concentration with time at a particular region is proportional to change in concentration gradients at the location of the system. Time lag. This is time required for diffusion to establish a uniform concentration of gradient within the membrane that separates the donor and receptor compartments representing T1. This is given by the following equation. T1 equal to h square by 60. H is the membrane thickness, D diffusion constant and T1 is time lag. Steady state. It is considered the diffusion originally dissolved in the solvent in its free hand compartment of chamber shown in the figure. Solvent alone is placed on the right hand side of the barrier and the solute or penetrant diffuses to the central barrier from the solution to solvent slide. In diffusion experiments, the solution in the receptor compartment is constantly removed and replaced by fresh solvent to keep concentration up to very low. This is referred to as shrink conditions, the left compartments being the source and right compartment the sink. In this diagram, we can represent the concentration gradients of diffusion across the diaphragm of diffusion cell. It is normal for concentration curve to increase and decrease sharply at the boundaries of barrier because, in general, C1 is different from CD and C2. Is different from CR. Originally, the diffusion concentration will fall in the left compartment and rise in the right compartment until the system comes to equilibrium based on the rate of removal of diffusion from sink and nature of the barrier. When the system has been existent a sufficient time, the concentration of diffusion in the solution at the left and right of the barriers becomes constant with respect to time, but obviously not to the same compartments. Permeability coefficient. This shows rate of permeation of drug molecules across semi-permeable membrane. Burst effect. It may be controlled release formulations immediately upon placement in release medium. An initial large bolus of drug is released before the release rate reaches the stable profile. This phenomenon is known as burst effect. In many of the controlled release formulation, immediately placement of release medium and initial bolus of drug is released. It is called as burst effect. Initial state of drug released into the receptor sites as a higher rate than the steady state release rate. Factors affecting diffusion: size, shape, concentration, temperature, charge, lipid solubility. First, size. Small molecules can easily slip into the polar heads of the phospholipids and through the membrane to other sites. So, small molecules can easily slip through the membrane than other molecules. Very large molecules cannot diffuse at the membrane. Shape. Glucose is able to get into the cell faster than other sugar made by facilitated diffusion. A carrier protein specific for glucose combines the outer surface close around it and then it opens inside the cell whether glucose is released. The carrier that returns to its original shape is ready to transport another glucose molecule. Concentration. Greater the concentration gradient between outer and inner of the cell, greater the rate of diffusion. So, when concentration is higher, diffusion is higher. If the concentration of oxygen outside the cell increases, then it will diffuse more quickly into the cell. Concentration of oxygen outside the cell increases, get more diffusion. Temperature. Increase in temperature causes all molecules to move faster. Diffusion is a passive movement of molecules, so quicker the molecules movement translates quicker diffusion. So due to increase in the temperature, the particles move suddenly. So when temperature increases, the more diffusion will take place. Charge. Ions or molecules within the charge cannot pass through lipid bilayer or diffusion. Other mechanisms involving protein carriers and ATP energies are required. Example: sodium, potassium, ATPase pump. Lipid solubility. Lipid soluble molecules can move through the lipid bilayer. These molecules are other lipids. Example: steroid hormones like testosterone and estrogen. Next, we are going to see about dissolution. Dissolution is the process in which a solid substance solubles in its given solvent. Diffusion means the movement of solute particles from higher concentration to lower concentration. Here, movement of solid substances dissolves, that is solubles in the given amount of solvent. Dissolution is the rate determining step for hydrophobic, poorly aqueous soluble drugs. It is a rate determining step for water hating group and the drugs which are not soluble in the water. Example, grisofluin, spironolactone. The rate of dissolution is given by Noyes and Whitney. dC by dt equal to K into Cs minus Cb. The equation is based on Fick's second law of diffusion where dC by dt is dissolution rate of the drug, K dissolution constant, C is concentration of drug in stagnant layer, B bulk of the solution at time t. Modified Noyes-Whitney's equation. Brunner incorporated surface area A in Noyes and Whitney's equation. dCt by dt equal to Ka Cs minus Cb. Afterwards, Brunner incorporated Fick's law of diffusion, expands its given equation to include diffusion coefficient d, thickness and stagnant diffusion layer heights, and volume of dissolution medium V. Factors affecting dissolution. The effect of agitation, effect of dissolution fluid, influence of pH in dissolution fluid, effect of surface tension of dissolution medium, effect of viscosity of dissolution medium, effect of presence of unreacted and reactive additives in the dissolution medium, volume of dissolution medium, sink condition, deaeration of dissolution medium, effect of temperature of dissolution medium, effect of agitation. The relationship between intensity of agitation and the rate of dissolution varies considerably to the type of agitations used, degree of lamellar and turbulent flow of the system, shape and design of the stirrer and physiochemical properties of the solid. Effect of dissolution fluid. Selection of proper medium for dissolution test depends largely upon the physiochemical properties of the drug. Influence of pH. Change in pH exerts greatest effect of drug solubility. For weak acid, dissolution rate increases with the increase in pH. For weak bases, dissolution rate increases with the decrease in pH. Effect of surface tension of the dissolution media. According to diffusion film theory, dissolution is the body drug governed by the interplay between release of the drug from solid surface and it transferred throughout the bulk of the dissolution rate. Addition of surfactants below CMC can increase significantly the dissolution rate because of the better penetration of the solvent into the tablet, resulting in greater availability of the drug surfactant. Effect of dissolution medium. If the interaction of the interface occurs faster than the rate of transportation, such in case diffusion control dissolution parameters would be expected, the dissolution rate decreases with the increase in viscosity. Effect of presence of unreactive and reactive additives. Example, when neutral ion compounds like sodium chloride, sodium sulfate and non-ionic compounds like dextrose is added to the dissolution medium, the dissolution of benzoic acid was linearly dependent upon the solubility in the particular solvent. When certain buffers and bases are added to aqueous solution, it increases the dissolution rate. Thank you.